I'll switch gear a bit. I don't have any quotes from philosophers or historical personalities, but I would like to talk about personalized medicine in autoimmune disease. And I hope to convince you that there is something that sets autoimmune diseases apart from other diseases when we talk about personalized medicine. The importance of personalized medicine in cancer was stressed in previous talks, and I believe that we can follow the same paradigm as cancer when we're looking at autoimmune disease that will allow us to much more precisely personalize diagnosis and hopefully treatment of people with autoimmune disease. As a paradigm for autoimmune disease, I've been studying type 1 diabetes for many years. We just started also applying the same paradigm to celiac disease, but I'll only talk about type 1 diabetes. So type 1 diabetes is very different from the diabetes most people are talking about. It has nothing to do with overeating or not exercising enough. It's an autoimmune disease. It starts very early in life. And it involves destruction of the cells that make insulin, the beta cells in the pancreas, by the immune system. Specifically, T lymphocytes is a T lymphocyte disease, which means that it's not due to antibodies. It's due to the T lymphocytes getting into the pancreas and actually directly killing the beta cells. It's very antigen specific. Only the cells that make insulin, the beta cells, are killed. The alpha cells that make glucagon and the delta cells that make somatostatin remain intact. So it's very antigen specific. Here is a healthy islet of Langerhans. This is the endocrine part of the pancreas, and this is the exocrine pancreas next to the digestive enzymes. And this is an islet that has been half destroyed by infiltrating lymphocytes. This is a process that lasts for months or years before any symptoms appear. So it's important to predict and try to prevent before it happens. By the time the blood sugar goes up and the child starts urinating and drinking a lot, it's too late. Most of these islets have been destroyed. So the cause is not fully understood, but we know genetics play an important role. I've spent much of my career discovering some of these genes and studying their effects. And here is the contribution of the virus known genetic loci. And how much of the lambda S is explained. The lambda S is the relative sibling risk. If you have a sibling with type 1 diabetes, you are 15 times more likely than the general population to get type 1 diabetes. And that 15-fold, of that 15-fold, 3-fold already, almost half of it, if you square 3.5, you get almost all of the lambda S of type 1 diabetes is due to HLA. And in the insulin gene itself also plays an important role. I don't have time to expand on this. And PTPN22, this is a lymphocyte phosphatase. So those three genes explain a good part of the sibling relatives. Essentially, is a good measure of the heritability of the disease. All the other loci, we started with 1,000 children and 2,000 controls. And our latest meta-analysis, we had 6,000 type 1 diabetes patients and 10,000 controls to get down to these genes that explain a tiny, tiny part of the heritability. We could come back with 100,000 cases, 100,000 controls, and we get genes with even smaller effects. But at some point, we have to ask ourselves, how can we use this information to personalize diagnosis and treatment of type 1 diabetes? If we look here, the HLA effect is known. Of course, HLA is probably the most important family of genes in autoimmunity, not only because they have a very important function to present antigens to the T lymphocytes, but also because they're the most polymorphic proteins in the human genome. So we've known about HLA for 3 decades, and we still have not particular applications on personalized medicine. A huge effect known for 3 decades. A few years ago, we had, in collaboration with Jerry Nippon's lab in Chicago, in Seattle, we found one epitope of proinsulin, of the proinsulin molecule, which is the main antigenic target in this autoimmune process, that is restricted for DR3401 as one particular HLA genotype, and that was 10 years ago, and we still there still has been no progress on how we can use information about this particular genotype in antigen-specific intervention. But that's still one promising way of using germline genomics. Insulin is also important. The insulin gene itself, there's a polymorphism that affects insulin expression not in the pancreas but in the thymus. And the thymus makes tiny amounts of insulin to tolerize the immune system. And there is a randomized prospective controlled trial to give oral insulin to desensitize the immune system, but only to people who have high insulin antibodies. So these are some examples of how we can use germline genetics to personalize medicine. But if we go beyond those 3 important genes and we start looking at those tiny effects, I don't see much prospect about this. So this is just describes how this genetic association started. Then we'll just compare the frequency of various sequence variants between healthy people and people with type 1 diabetes. Another possible explanation for why some people get diabetes and most people don't is environment. People talk about viruses. There's a lot of talk about foods, introduction of cow's milk, gluten. None of this has been proven. So we don't know what the role of the environment is in type 1 diabetes. But we know that genes are not enough. The genes we inherit from the parents. If we get identical twins, if you have a twin with type 1 diabetes, you have a 50% chance of getting type 1 diabetes yourself. So this is the first twin, age of diagnosis of the first twin, age of diagnosis of the second twin. See, it plateaus at about 50–60%, a little higher for antibodies without having the actual disease, but it's never 100%. And even when the 2 twins are concordant, there could be decades of difference between the first twin and the second twin. Now that doesn't explain; it's not explained by genetics. Identical twins have exactly the same genome. So what happens? Environment could be an explanation, but most people who have type 1 diabetes have the disease before they're 18. At that age, most pairs of identical twins live in the same house, eat the same food, are exposed to the same viruses. Not a satisfactory explanation. Even more striking, the NOD mouse is an excellent model of spontaneous autoimmune diabetes, just like in humans. Male NOD mice only get 50%, only get diabetes 50% of the time. They're identical twins, essentially. There's one patient cloned in thousands of copies. They live in the same cages, eat the same food. Why half of them get diabetes and half of them don't? There must be some stochastic event, and one stochastic event is spontaneous mutations in the soma after fertilization. So somatic mutations, just like in cancer. And the New England Journal papers were mentioned that as people get old, they get mutations that are connected to leukemia without getting leukemia. And this is on immune cells that obviously have an impact on autoimmune disease like type 1 diabetes. So based on information like this, we formulated this hypothesis that we have billions of T cells in our body, but the disease, the autoimmune disease, is going to be caused only by 5, 10, 20 cells that recognize insulin as the antigen, and then multiply, expand, and make millions of progeny. And when the cells expand like this, somatic mutations can happen, just like in cancer. It's a paradigm very similar to cancer. We have a bunch of lymphocytes here. You can divide them into subtypes based on what cytokines they make, whether they're aggressive or protective. But here you have a bunch of cells that have different T cell receptors. They recognize different antigens, and only a tiny fraction of any subset of lymphocytes will recognize self-antigen, and for some reason will not have been eliminated by the self-tolerance mechanism, and they will proceed and then will start multiplying. But then there are other self-tolerance mechanism that prevents them from multiplying further and further. Suppose that as the cells multiply, the undergoing somatic mutation that allow them to bypass the first roadblock that the immune self-tolerance system puts in front of them. So they go to the next phase, and then as they multiply, they can have another somatic mutation and so on. A paradigm very, very similar to cancer, except that what is inactivated here is genes that have to do with self-tolerance, not necessarily genes that have to do with control of proliferation, which is what happens in cancer. So here in this situation, we have a very sophisticated subset of cells, and that's 90% of research that's been done on type 1 diabetes are looking at subsets of cells. But within this subset of cells, no matter how elegantly and how sophisticated the cytokine profile is, only a few cells will go through this. And these are the cells in our hypothesis that are causing autoimmunity because of postzygotic mutations. So remember, the New England Journal papers looked at whole blood. They didn't look at specific lineages, at specific clones of cells. So in collaboration with Brent Richards and Tim Spector in the UK, we looked at the Twins UK project. These are identical twins. And one idea of getting how many mutations happen in blood that have high enough mosaicism to be detected in whole blood. We look at discrepancies between monozygotic twins. So we look at 94 monozygotic twins that have been genotyped for 600,000 SNPs throughout the genome. And most of these were sequencing errors, but we were able to confirm 2 out of 80 with Sanger sequencing. These were real, identical twins in whole blood, had somatic mutations at high enough mosaicism to flip a genotyping call. And we confirmed this by direct sequencing of the blood of these twins. Because 600,000 SNPs is a lot, but they represent only 1/6,000th of the genome. So with simple arithmetic, we can calculate every person carries in their blood something like 600, and then this is just blood cells. We're not talking about other cells in the body. About 300 point mutations. And some of them could promote proliferation and survival, and that's why they reach levels of mosaicism high enough. Because when you have high mosaicism, there's two explanations. One explanation is that the mutation happened very early after fertilization. But very early after fertilization, there are very few cells. The probability that a mutation happens at these early stages is very small. Most mutations happen later when there's millions, billions of cells. It's much more likely that the mutation will happen, and then only a small part of the cells in any organ system will have these mutations. So very low levels of mosaicism. But then when this particular lineage, I'm trying to point with, okay, with this particular lineage, highlighted in green, reaches a level of mosaicism that detectable in whole blood, means that this cell has had a proliferation advantage. And not only point mutation, but also copy number mutation, big deletions or duplications happen very often in the blood of individuals, and can be found up to 2% of the population after the age of 50. So if it's age dependent, it means the high mosaicism is not because the cell underwent the mutation early in development, but because these mutations give some proliferation advantage, and that's exactly what would promote autoimmunity. We looked at, we have 1,000 kids with type 1 diabetes whom we tested, we genotyped them with SNP arrays for our genome-wide association study. This is 500,000 points in the genome where there is a common polymorphism. And when we looked, went back and looked not at the finished genotype results, but at the raw fluorescence. If we add the fluorescence for both of the alleles, you get something like this. This is a logarithmic scale, so the two alleles should have a ratio of 1 if you're heterozygote. So the average is 0, and there's some noise around 0. That's the total fluorescence that we call LRR, and here is the ratio of the fluorescence between the two alleles. So these are the heterozygotes. The ratio should be 0.5. The homozygotes for one or the other allele, which are at 0 or at 1. And in some part of the genome, you see this. You see the raw fluorescence going up. Not only this, but also the 50-50 ratio between the two alleles gets disrupted, which means that we have a duplication here. And in this particular diabetic child, the duplication involved the gene that's very important in stem cell, in hemopoietic stem cell proliferation. But it was only 2 out of 1,000 kids that we found this. But this was whole blood. It requires a very, very high level of mosaicism to detect in whole blood. We decided to look at specific lineages, that the ones that go in the pancreas and destroy the beta cells. So those in blood are very low frequency, maybe 1 in 10,000 cells. You'd have to get pancreatic biopsy to get to those cells, which of course our ethics committee wouldn't be terribly excited about permitting. So we decided to look at blood, to look at the pancreas using the NOD mouse model. So we took NOD mice as soon as they became diabetic, and we dissected the pancreatic lymph nodes, and they compared the DNA of memory T cells to the tail clip DNA. DNA in the tail were assumed to represent the germline, what this mouse inherited from its parents, and the memory T cells, which is this fraction. And we concentrate on the CD4 cells for a number of reasons. Cells that have already replicated upon contact with the antigen. And some mice, actually, in some mice, we found that pancreatic lymph nodes contained only memory cells. And these were the more interesting findings that we found in these mice. And in some mice, as you would expect, the CD4 cells, memory cells, were only a small part of the total cell number. So we extracted DNA from those cells and from cells representing the germline, and we look for copy number changes with a standard method, which is CGH array. We decided to start with copy number changes, not because point mutations are not important, but copy number changes are much easier to identify at low level of mosaicism, and also much easier to figure out what they do functionally. So, so you take the test sample, which is the lymphocyte DNA with one color fluorescence, and the reference sample, which is the tail DNA with a different fluorochrome, and you run them on an array. And whether there is copy number change, this is reciprocal, actually. You see deletions and insertions labeled with the different fluorochromes. You can look at the ratio of the two fluorochromes for each probe, and the probes cover the genome equally. So first of all, we look for some technical validation. We know that lymphocytes rearrange their T-cell receptor. This is a very unique property of lymphocytes that no other cell does. The cells of the tail don't do. So in the places encoding for the beta, gamma, or alpha chain of the T-cell receptor, we see a clear deletion. Here, normally the signal is 0, and you have a little bit of 2 copies of the gene corresponds to 0. A little bit of noise around 0, but then here clearly a deletion. So these are built-in positive control. So we know that the method works. And then what we did two strain of mice that have known CNVs, copy number variation, and we were able to detect very easily the copy number variations, even when we had mosaicism only 25%. By mixing DNA from the two strains, we could imitate mosaicism down to the 25%. And it's clear that our method can detect mosaicism much less than 20%. So this is just the methodological aspects. In the results, actually, we have 12 mice here. We've done more since then, but the results are the same. So we find a number of copy number changes. Here on this line, you see this mouse had 1. This one didn't have. We couldn't find any. 3, 18. And then we had 2 mice that have a very large number of mutations, 208 here. And these were the 2 mice whose pancreatic lymph nodes contain only memory cells, and there were no naive cells. So this was almost like leukemic cells. So there's a very, very large number of copy number changes. This mouse is going to die in a few days, even if we didn't kill it, because of the diabetes. But if we left it alone, I'm sure they would get some kind of lymphoma. And if NOD mice survive for a long time, sometimes they get lymphoma. What's more important was a very considerable number of mutations, copy number mutations, that happened independently in different mice. In cancer, this independent occurrence in different individuals is the hallmark of a tumor suppressor gene, a gene that's important enough for the disease that it is mutated independently, somatically, in different mice. These were the 4 mice that had their pancreatic lymph nodes taken over by memory cells, and 2 of them had the largest number of copy number mutations. I'll just show an example here. Here's the 2 histone H3 gene clusters on mouse chromosome 13, and I have 2 different mice that had multi-copy amplification of those histones. Now, those histones play a very important role in DNA replication, and that's when somatic mutations happen during replication. So if you mess up the system by overexpressing those very important molecules, then that explains why the rest of the genome in those lymphocyte lineages was all over the place, like a leukemic cell. This is an example of TMEM, another 107, a gene that has not been studied. We don't know what it does. 3 different mice had multi-copy amplification at high mosaicism level in their pancreatic lymph nodes. So it is like cancer, except that the mutations don't cause leukemia, just like the New England Journal mutations found in normal people, but that are enough to cause autoimmune disease. And we confirmed these copy number changes with MLPA, which is the gold standard for CNV diagnostics. And then as a negative control, we infected mice with Leishmania major. It's a nasty bug that causes inflammation in the foot of the mouse, and we put popliteal lymph nodes. And now we have T cells that are doing their normal job. They're not causing autoimmune disease, but they're doing their normal job, and we find no copy number changes when the T cells are doing their normal. It's exactly the same inflammatory environment, but it's adaptive. Okay, for human studies, we have to use blood, so to get around the point that in the blood, those cells are very rare, we culture them for 2 weeks with proinsulin, which is their antigen. And when T cells see their antigen, they start multiplying. And then we stain them with a dye called CFSC. Every time the cell divides, there's less of the dye in the cell. So cells that have lost their dye means are the cells that have multiplied a lot. And that's what we see here. This is CD3. So these are the T lymphocytes. These are other cells in the blood. T lymphocytes, this is CFSC high, the cells that have not multiplied. And this is CFSC low. It means the cells have lost their dye. These are the cells that see insulin and attack the pancreas. This is the blood of a child, a 70-year-old child just diagnosed with type 1 diabetes. Our positive control with tetanus toxoid, of course, this child, like everybody else, has been vaccinated against tetanus. And media alone, we don't see this cell proliferation. And we did the same thing. We compared the cells that have multiplied after exposure to insulin with cells that have not multiplied after exposure to insulin. And we see we look for copy number changes that are specific for those cells. We also sequence the T cell receptor to get some idea of clonality, and we see the highest clonality was 19%. It means 90% of these proliferating cells are descendants of one single cell. If you can imagine how many proliferations must have happened, and so there's a lot of opportunity for somatic mutations. So when we looked at those human subjects, we've done 6 children so far with type 1 diabetes. And you see that some of them, that all of them have copy number changes. And one of them had a very large number. The child doesn't have leukemia and probably will never get leukemia, but it was enough to cause diabetes. And more importantly, we see overlaps, the same mutation happening, the same 4 mutations happening in at least 2 of these children. So again, in the paradigm of cancer, that's considered very strong evidence that that these are causal. This is just not mutations that happen at random because the cells proliferate a lot. But if it happens again and again in different individuals, they must mean something. So we have here Wnt11. This is one good example of a gene that has overlapping. We have 3 different children here. 2 of them have a deletion that completely destroys the gene, and one of them has a duplication, but the breakpoint of the duplication are in the middle of the gene, which means although the DNA is duplicated, the gene is inactivated. So 3 out of 6 children, 50% of type 1 diabetes, if we can extrapolate from these small numbers, have mutation of Wnt11. The other WNTs have been studied. WNT11, we know nothing about it, but I bet you is a very important gene in immune function. And here is another example, another gene that has not been studied very well, but the little bit that's known about this gene shows interaction with the known immune players. One deletion and one duplication here overlapping on the same gene, both of them deleterious to the gene. So these are just some examples. So in conclusion, we've shown that there are somatic mutations that happen in those lineages of antigen-specific cells that cause autoimmunity. What we find more exciting is that these mutations happen in just one cell lineage. They haven't had a chance to be purged out by evolution. They don't need to be compatible with survival of the whole body. They must have much stronger effects than the germline mutation. What you inherit from your patients have been through thousands and thousands of generations of evolution. If it's something that will predispose you to autoimmune disease, it will wipe it out, especially type 1 diabetes. You die before you can reproduce. But those somatic mutations don't have these constraints, so we expect there will be much, much stronger stronger biological effect. And of course, we can, instead of making a germline profile, which has a lot of problems in terms of predicting and preventing disease, now we can have a somatic mutation profile, just like in cancer. In cancer, this whole idea of genetic profiling is much, much more powerful because, exactly because those somatic mutations have not gone through evolution, have much, much stronger effects. So much of this work was done by my student Mahal Rayami. She's doing a PhD in my lab. Li Chuan was the bioinformatics person, my postdoc who did, and the other people in the lab contributed. Here's some of the people at the organizations that funded the research. Thank you.
Thank you. Very nice presentation. I have two questions. The first is specifically related to the etiology of what activates the T cell population, and you identified as a CD4 population of cells. T cells usually require, in order for antigen presentation for them to become activated, macrophages. What role does the macrophage have in the alteration such that the activated T cells then become autoimmune?
Well, that's a very good question. Macrophages are not expanding like T cells, so I don't think that somatic mutations in macrophages will be important, but of course they are very important. As you know, they're polarized into M1 and M2, and M1s activate lymphocytes, M2 actually are anti-inflammatory. So obviously they play very important. The third of the germline loci that I show, that's PT-pen2, it's a lymphocyte phosphatase. Everybody calls it lymphocyte phosphatase, but actually is expressed at very high levels in macrophages. And I have a student doing a PhD in my lab on the effect of this lymphocyte phosphatase in macrophages. And she finds differences in how good they are, depending on the genotype they have at PT-pen22, in stimulating T cell proliferation, both in human blood and in NOD mice. So, yeah, macrophages are important, and they're definitely modulated by germline genetic variants. Whether somatic mutations in macrophages play a role, that would be much harder to evaluate because they don't clonally expand the way T cells do.
Thank you. My second question addresses potential therapies. Since this activated T cell subpopulation of CD4 as a blood-derived stem cell, number one, but number two, also to address beta cell regeneration, what role do you see, if any, for stem cells in utilizing as a stem cell transplant, both to replace the defective T cell population, but also importantly, perhaps, maybe even then also engineer the stem cells, maybe even through iPCs, to regenerate beta cell replenishment?
Well, I'm not a stem cell person. I can't say much about regenerating the beta cells in the pancreas with stem cells, but there's obviously a huge international effort trying this. In my mind, it's futile unless you get a good hold of the autoimmunity. If you regenerate the beta cells, they will just get redestroyed. But to replace the defective T cells, that's a much more attractive proposal.
We can repopulate and
Yeah.
replace the hematopoietic system.
Marrow transplant has been proposed for treatment of the autoimmune disease. It's been tried in multiple sclerosis, where you have no other alternative. In type 1 diabetes, we can give the patient insulin, and they can live a good life for decades. It would be a bit drastic to do marrow transplant, but if you could identify some of these duplicated cells, genes, are surface protein, you could use flow cytometry to isolate those cells and get rid of them somehow, or use an antibody, a cytotoxic antibody against them. These are all very attractive prospects. In tomorrow's talk, I'm going to talk about T cell receptors. The other idea is those cells, okay, they've expanded. I don't care what genes drove their expansion, but the one thing they have in common is they like insulin. So is there any way we can isolate the cells based on the T cell receptor, recognize the insulin, and then hit them with a specific antibody? That, again, is not very simple, as I will give you all the information in tomorrow's talk. Thank you.
I just wanted to ask you, you mentioned in one of the slides about the environment. You know, that is very
Your favorite
issue
topic.
now with all the
Yeah.
diseases, you know, and you showed some substances, you know, that would be influenced. What do you think? Can you predict, you know, what the future look like? I showed in one of the slides, you know, this small man, Laron syndrome. They never get diabetes, you know, so it would be very interesting to speculate that one could find out, you know, what is happening.
Well, the T lymphocytes have a growth hormone and IGF-1 receptors. So obviously, if you have very low IGF-1 levels, it can alter the immune system. But in terms of food, there's been a lot of noise about early introduction of cow's milk and breastfeeding and all that. A prospective trial to eliminate cow's milk in the first 6 months of life didn't show anything interesting. So I don't believe it's cow's milk, but gluten now has surfaced as another possible trigger for the autoimmunity. Type 1 diabetes and celiac disease have a lot of things in common. It's the exact same HLA genotype that predisposes to both. So early introduction of cereal is now being tested as possible. There is a so-called TEDdy study. It's an international collaboration to follow newborns at high risk genetically with a risk HLA for type 1 diabetes. Follow them for years until some of them will get diabetes, and see what they were eating, what viruses they were exposing. There's stool collections for microbiome, and I'm sure something interesting will come out of it. It's a huge effort because you have to wait until something like 3% of these kids will get diabetes.
Just a quick question, actually. Well, maybe a quick question. I think the main message that I got from your talk was that the diabetes 1 is not just caused by the insulin. It's the accumulation of many mutations in different genes. And as you mentioned, it's amazing. It's at one lineage of cells. So do you have in mind any mechanisms for these mutations? I mean, there's some specific genes in DNA repair involved that you can
this is a very good question. We didn't see any genes involved in DNA repair. If they did, they'd probably get cancer, not diabetes. But in the NOD mouse model, we saw this striking phenomenon. 3 different mice had multi-copy amplification of histone 3. Histone 3 is crucial for DNA replication, and if you mess up DNA replication by overexpressing one protein that controls it, you're more likely to get mutations elsewhere in the genome. In terms of how mitotic accidents happen that cause mutation, that's, this is a huge literature on which I'm not particularly expert. I don't think it's any different in this particular setting. Of course, we know that parts of the genome that are much more likely to undergo copy number changes than others. If you have a sequence that's flanked by repeats that are homologous, you have no homologous recombination, which is the most common cause of copy number changes. But interestingly enough, that's something I didn't have time to talk about. In T lymphocytes, T and B lymphocytes are the only cells in the body that mess up with their DNA on purpose. The T cell receptor and the immunoglobulin receptor, in order to recognize the millions of antigens that we're going to face in our life, they have to generate diversity. And the diversity, I'm going to talk a lot about it tomorrow, is generated by cutting and pasting different parts of the T cell receptor and the immunoglobulin gene. So this is the only known example of a cell messing up with its own DNA. They have a particular enzymes that do this, the RAG enzymes, and the RAG enzymes go to those to the receptor gene because they recognize a specific sequence. In T cell leukemia, like any other leukemia, you see a lot of deletions and duplications in the leukemic cells, and half of them are on genes that are flanked by sequences that are not exactly the same as the RAG recognition, but similar enough to the RAG recognition sequences. So this is a very attractive way of explaining why T cells may have a higher rate of mutation. Actually, we're looking into this. We're doing the bioinformatics to see if the same, and I'm sure we'll find the same thing here, that these genes are mutated repeatedly because they have RAG sequences.
Okay, thank you. Any other question? So I would like to invite Professor Vincent to